Hello, everyone. My name is Steve Shih, and I'm currently an assistant professor out in Concordia University in Montreal, Canada. It gives me great pleasure here today to speak to you about some of the work that my lab has been working on, specifically in the areas of gene editing, microfluidics, and automation. And so the title of my talk today is Gene Editing by a Click of a Button. And the way I sort of designed my talk today is through these learning objectives, specifically three of them: defining current paradigms of gene editing, identifying bottlenecks in the process of gene editing, and then I'll sort of describe some of the solutions that we came up with to automate approaches towards gene editing, specifically in the area of microfluidics. And so what I'll do is I'll start with defining current paradigms of gene editing. And for those of you that don't know how sort of gene editing works, the idea is that you essentially want to edit a specific gene that you want. Essentially, what you have is this Cas9 enzyme or protein that's kind of shown here in the yellow. And the special part about this Cas9 protein is that it acts like a molecular scissor that will cut the DNA, essentially the one that you actually want to edit. Now, the special feature, though, with this sort of Cas9 is that it just won't cut anywhere. It actually recognizes short DNA sequences called PAM, as you can sort of see the label there on the screen, and it serves as a tag. And at the same time, it actually sits adjacent to the DNA sequence that you actually want to cut out. Now, to actually guide the cutting or guide the Cas, you have also something called a guide RNA, which actually binds to the complementary DNA target that you actually want to cut. So essentially, gene editing, if you want to perform it in your own lab, all you need is the actual protein, the Cas9, and the guide RNA. But once you actually have it cut, you create this double-stranded break. What usually happens actually in the cell, and the problematic sort of thing that happens is that the cell's natural repair mechanisms start to kick in and starts to introduce these mutations. Now, one of the ways that they try to introduce mutations is through this non-homologous end joining technique, which essentially is nucleotides being inserted or deleted into sort of the regions that's been cut. And this essentially induces any sort of mutations or errors and essentially disrupting gene function overall. The other way, where it's more controlled, where we have this homology-directed repair mechanism, and it's essentially filling the gap with a sequence of different nucleotides. And usually this is a short strand of DNA that acts as a template, which can be supplied by usually the scientist who wants to perform the edit. And this can overwrite sort of any mutations or incorrect errors that are represented after the double-stranded break. And this whole system of CRISPR-Cas, now you can essentially use these methods to perform gene knockouts, knock-ins, precise base editing for a number of different applications, like diagnostics is very popular, generating or producing different cell lines, and also deciphering different pathways like cancer. But gene editing, I mean, it's taken off so quickly, but essentially, what is the main challenge that we have with gene editing? Now, I would say to explain this, I want to first go through the process of gene editing. And so essentially, when you want to start gene editing, the first step that you would do is that you want to select essentially a cell line that you actually want to edit the genes. Then you want to design the construct, or specifically the guide, as I explained before, that will target the gene that you want to cut. Next, you want to actually deliver this construct into the cells that you want to edit. And then this can be done with a number of different techniques, such as electroporation, liposuction, squeeze techniques. Then once you have incubated, the cell has sort of taken over its mechanisms and then performed the actual edit itself. Then what happens is that you need to perform some sort of selection technique, like antibiotic selection or FACS, and then sort to obtain the cells that actually have been edited. But once you sort of gather this homogeneous populations, then you want to obtain the actual single clone. And that's usually done with some sort of constant pipetting, using techniques like limited dilution or colony picking techniques. And then finally, You can get that single clone, place it into a well plate or some sort of platform like a well plate. You can expand the cells and then use these expanded cells to sort of validate if the editing actually occurs. But having all this, the main challenge still is what is that? The main challenge here is that the techniques actually that are involved for gene editing actually needs to be optimized. Transfection, selection and sorting, single cell cloning, validation, and expansion, all these different steps all require different technologies to actually perform their tasks. And this actually contributes to a lot of variability in the results, especially between different labs that are performing the same type of editing. And this really poses many challenges to really fully automate these processes, particularly around, transfection efficiencies, enriching and recovery of single isoclones. And because of this, this pipeline can take months at a time for you to get the actual single clone or the edit that you actually want. It can incur much higher costs. And at the same time, it requires frequent manual intervention. And so what we propose in my lab, or what I'm going to show you, is that microfluidic systems can actually miniaturize these experiments. You can multiplex them in parallel, reducing the cost, increasing the throughput. But the main advantage is that we're able to integrate and fully automate these gene editing steps into sort of one platform. And the technologies that I'll focus on today, I don't have a lot of time to discuss all the different types, but I'll focus mainly on the droplet-based technologies, where mainly the droplet and channel technologies, the digital-based microfluidic technologies, and I'll also combine both technology, which we refer to them here as hybrid microfluidics. So now what I'll do is I'll take some time to describe both these technologies, starting with digital microfluidics. For those of you that are not familiar with digital microfluidics, essentially it's a liquid handling technique where we manipulate droplets on an array of electrodes. A unique feature with digital microfluidics is that you can manipulate droplets on an open or closed face surface. And as you can see from these images here, you have lots of or have the opportunity to control these droplets, has this individual addressability, which allows you to dispense these droplets into smaller droplets with excellent precision. You can move them, you can merge them, you can mix them and split them as well. But how does digital microfluidics all work is that essentially you have the side view of a device. You have these metal electrodes that are patterned on a glass substrate. You deposit an insulator, a hydrophobic layer, and then you would put a droplet on top of the surface, which is now then covered with a top plate counter electrode, which contains also a hydrophobic layer. Now, typically you can manipulate the droplets in air, so nothing surrounding the droplet, but you can also have some sort of filler fluid, such as oil, which sort of minimizes sort of the friction between the droplets and the surface for the actual droplet movement. And if you want to go into more detail, essentially how it works is that you have these electrostatic forces that are coming into play. Essentially, it's like if you have like a comb that you're brushing your hair, static electricity starts to develop. And if you bring it to a stream of water, you can start to see it bend towards the comb. And so by using this sort of physical phenomenon, you can essentially switch on and off these different electrodes, and then essentially position these droplets anywhere on the array. Now, one of the main advantages, though, I would say with digital microfluidics is that you have this amenability, this opportunity for automation. There's no valves or pumps here as you would have with traditional base microfluidics. But what you have with this automation system is that you have this matrix board of containing switches, and these can be essentially directly connected to the device itself so that you can turn on and off the different electrodes, as I sort of explained previously. Now, since this is sort of an automation conference, I'll kind of show you in more detail what this automation system really consists of. Essentially, you have this user interface that controls a microcontroller to control all the switches that I showed you before. And then you can also control all the voltages, the frequencies that are needed to actually manipulate the droplets. Now, if you are using traditional base microfluidics, you can also include syringe pumps as a plug-in into the system. But essentially, all this sort of different parts can be sort of placed into essentially a box that can be controlled by a software interface that we have created in our laboratory. But anyone can actually create their own software to control this hardware system as well. Now, the software that we have created is that you can have your device design essentially on the screen, where essentially it matches the electrodes on the device, and then you can press or create sequences such that you can activate the different potentials on the device. And then you can also create specific functions, and you can create those dispensing functions, mixing functions, merging functions of the droplet, and essentially this all be translated all into moving the droplets onto your device. And if you're interested in learning more or wanting to set up your own microfluidic system for automation, you can find a lot of the code and the software and how to build the hardware on our Bitbucket website shown here. Now, I'll just talk briefly about droplet and channel microfluidics. As you know, digital, you're working with array of electrodes, but here you're putting droplets inside the channels. You have a two-phase system that usually consists of an aqueous flow and an oil-based flow. And then you can essentially create droplets through this T-junction shown in the left image here. And then you can have this hydrodynamic focusing where you can create many droplets as well, shown in the right image here. Now, I would say some of the differences between digital and droplet is the following, is that in a lot of droplet and channel papers or in the literature, you'll see that they have very high throughput. You could probably generate 1,000 droplets at the same time. But often all these droplets here are manipulated in series. And so the key challenges I would see with droplet and channel is that there's a lack of control. If I wanted to generate 1,000 droplets and I want to access maybe the 100th droplet, that becomes very difficult. I would have to wait for all the 99 droplets to pass by. At the same time, droplet addition, it's very hard to add reagents, add media, add buffers, add drugs to the other with other droplets, and so it's really difficult to create a dilution series of droplets. But with something like adding the controllability that digital microfluidic offers and adding the throughput that droplet and channel offers, we can create some of these hybrid microfluidic devices, which now you have both the advantages of controllability and throughput. So here you can see from the images, you can essentially merge different droplets anytime you want to. And then here you can sort, two types of droplets, what I've shown here as green and red, but you can have many different sorting channels. Here I just show binary, but in the paper that we have from Fatemeh Ahmadi, we can sort many different droplets at the same time. So now I've sort of given you guys some ideas about gene editing, what are sort of the bottlenecks in the process of gene editing. Now I'll sort of go into some of the automation or microfluidic automation approaches on how we can actually gene edit some of these cells. Now, before I do that, I just want to explain that there's sort of a mismatch between microfluidics and cell culturing. If you want to edit cells, the first thing that you need to do is that you need to make sure that you can cell culture. But if you've done cell culturing before, you know that cell culturing likes hydrophilic surfaces. But what I explained before is that digital microfluidic systems, they actually have a hydrophobic system. So because of this mismatch, there poses a challenge on sort of how we sort of culture cells and therefore how we can edit cells downstream. But during my sort of graduate school days, we were able to come up with a system where we create this hydrophilic site on the digital microfluidic device. So instead of having a constant hydrophobic layer on the top plate, we create this hydrophilic site. And by doing that, what you can see from the image on the left here is that you can create these miniaturized or smaller droplets on these hydrophilic sites. And then because of that, you can create these miniaturized droplets, but at the same time, you can have these adherent cells start to adhere onto the surface or the top surface on the device while allowing to manipulate the droplets using the bottom plate of the digital microfluidic device. So with this in mind, one of the first pieces of work that we did in my lab is to try to implement sort of the first steps of gene editing on digital microfluidic device. So here I'm going to try to explain to you the 4 steps that we've implemented to manipulate the droplets. So I'll go into sort of the device design, show you how we validated our device, and we did a proof of concept actual knockout. And then we also showed an application where we automate a proliferation assay. So I'll start with the device design. So what we have here, again, is that we're working towards automating this workflow where we have first starting off with the plasmid design. We perform the cell culturing, the editing, and the data analysis to see which ones have been edited and which ones have been knocked out, et cetera. And so here we would take these last 3 steps and integrate them on the digital microfluidic platform. And sort of here is the device design that we have. And it consists of 2 key features or 2 key components on the device. One, we have a region where we can prepare the lipid complexes for DNA or RNA or protein delivery. This will be used to essentially deliver the Cas9 and the guides directly into the cells. And then we also have cell culturing regions where we have this hydrophilic spot where we can allow all these cells to actually culture on the top plate. And so what we've done is that we took the timeline that is needed to edit the cells, and then we followed a number of different steps. So here we would seed the cells where we create droplets by dispensing them on the digital microfluidic platform. So you can see from the images here, on the left panel, you see like sort of the GUI interface that we have, and this is matching the droplet movements that you see live or images that are from a live movie of showing the droplet movements on the device. And so you can see the first step is we actually seed the cells. Step 2, we actually combine the lipids and the DNA so that we can actually deliver them to the seeded cells. Then we would incubate the lipid complexes to create the lipids with the DNA inside. We can dilute them as well to create many different concentrations. And then finally, we would actually deliver the lipid complexes to these hydrophilic spots. You can add drugs in the meantime, you can maintain them, and then you let it incubate such that you can determine the transfection efficiencies, or you can outgrow them, determine the knockout efficiencies, or you can actually continue to perform an assay directly on the device. And so the first step, what we've done is we actually took lung cancer cells as a test for the editing. Now, as you can see on the left panel here, or the left image, you see that these lung cancer cells are actually green. Of course, lung cancer cells are not usually green. So what we've done is we integrated this green fluorescent protein inside the cells. And then the first step, what we did was we created a guide that actually knocks out these green fluorescent proteins. Now, one of the first steps we did was we wanted to check if the viability and the morphology between our device versus traditional well plates, if there's any differences. And as you can see here, just from looking qualitatively of what the cells are looking like on the hydrophilic spot versus the well plate, we don't really see much of morphological differences, which means it's promising that we can sort of continue sort of going through the gene editing process downstream. So the next thing that we did was we actually performed the transfection on the chip. Now, one of the first problems that we faced when we were trying to do the actual transfection, as you can see, is that there's a high confluency. When there is a high confluency of the cells start to overlap with each other, which makes them very hard to count through imaging techniques. And especially if they're fluorescent, as you can see here, some are very dim, some are overlapping. So then we have this problem where we're unable to accurately count the cells. And this is a problem because downstream, if we want to look at efficiencies, we're not sure how many cells have actually been transfected, how many cells have actually been knocked out compared to the starting population on the device. And so a solution, what we did, or what we came up with, is that we created this automated pipeline to accurately measure the transfection and the knockout efficiencies. And so here's just a brief overview of our transfection pipeline. And essentially what we did is that we labeled the cells with Hirschstain, which labels all the nuclei of all the cells. And then we would look at an M-cherry image where when we transfected the plasmid, it has an M-cherry reporter. And by masking the objects together, we can then sort of calculate which ones have been transfected. And then looking at GFP, we can actually figure out which ones have been knocked out. So we did some preliminary experiments where, we created different dilutions of the lipid complex versus media ratio. And as you can see, as the higher ratios, 1-to-1, they are usually needed for higher or more efficient transfection on the chip. At the same time, we also compared if we tried different incubation times, and then we compared them to the standard technique of well plates. And what you can see here is after 48 hours, we achieved the highest transfection efficiency, around 70%. And this is promising, such that we're seeing very good similarities with the traditional platform on the well plates. So next, we want to sort of evaluate or show GFP knockout. So again, we just took or created guides that sort of or targeted 3 different locations on the GFP gene. And what we found is that efficiencies for both knockout on the well plate, the traditional platform, and our digital microfluidic device were very similar, and that we're achieving around 40%. And we used to get this number, we used the standard imaging pipeline for the analysis. So we have, again, the GFP, we stain the cells with Hirsch stain, we mask the images together, and you can see some of those cells that are there but don't have GFP, and then we count them as sort of a knockout. And that's how we obtained our efficiency. So then we went on to sort of perform this a microfluidic automation sort of proliferation assay. And what we did is that we wanted to evaluate the potential of using our platform for gene editing. So what we did is we explored this relationship between gene function and cell phenotype by studying a cellular pathway. Specifically, we looked at the ERK and MAPK, or the kinases pathway. And as you can see, the RAF genes and the RAS genes are upstream to these components of the kinase signaling cascade. And so therefore, because of this, they're essentially nodal points in cell proliferation, flagging them as potential or potent oncogenes. And so we use them here as essential targets. So the guide that we created would target RAF1, and then we also added a drug, something called sorafenib tosylate, that would inhibit the expression of BRAF and also RAF1. So what we did first is that we sort of looked at 4 different conditions. So we transfected cells under these 4 conditions. As already you can see, is if you provide genomic disruption, you can start to see a decline in sort of the cell viability. And so you see reduced proliferation. On the second day, you would add the drug. So what we did was we added sorafenib tosylate, and you can see further decline into the cell viability or the cell health for the ones with the guide and the drug. And then if you continue on to let it outgrow over several days, because we do see sort of a heterogeneous population, we do see some increase in the growth in some of the conditions. But the main point here is that with the drug and with the genomic disruption, you can start to see a much lower or increase in cell deaths and much lower in cellular viability. And we sort of prove this too by looking into this dose response curve where we had the inhibitor and also genomic disruption. And here we observed that genomic disruption, lower doses levels, are capable of reducing the cell viability. Essentially, what we're showing here is that the combination of genomic disruption and enzymatic inhibition leads to cell death of the cancer cells. So this is really nice, is that we essentially showed that we can automate the transfection, automate the cell culturing, the editing, and then finally automate the assay and also the analysis of whether if it's been transfected, automate the efficiency of transfection and knockouts as well. Now, if I refer back to the pipeline, so again, the problem arises to, we've done the transfection, we've done the editing, but now how do we actually achieve, selection sorting of the clone that we want, or, how do we isolate the single clone? How do we expand? We still have this heterogeneous population. So what do we do here? So how do we get to this sort of single clone? And so what we've done is we created a device, another microfluidic platform where it combines the droplet and digital. And we sort of use this idea to trap the cells or to capture the cells, have in situ droplet generation, and then actually select the clones that we want through imaging techniques. And so again, here's the sort of the platform that we use for selection and isolation. It's this 3-layer device that will provide the control, has this on-demand functionality where you can select the droplets anytime you want, create droplets anytime you want, And at the same time, we have throughput as well. And really all it consists of is the digital microfluidic layer, the dielectric for the digital microfluidic layer, a channel layer for the droplets, and then a PMS slab to essentially provide the reagents and as well prevent any sort of evaporations of the liquid. But just to remind you again, all this can be done with automation by just a simple interface with our graphical user interface. And essentially you can create sequences or functions that you can just essentially click and then be able to automate the droplet movements on our device. So to give you more detail, our device consisted of two parts. One part is that you can create this on-demand droplet generator. And so essentially here you can generate monodispersed droplets of different volumes on demand. And so we compared it to, the typical hydrodynamic focusing way of generating droplets. And you can see here, essentially you can have a range of volumes that you want with the on-demand generator. In the second part, we can essentially trap the cells, create the droplets in situ, release them or keep the droplets. And essentially using these sort of functionality, we can then isolate the single clones. Now I'm going to talk to, there's actually essentially 5 steps that we need to actually get your edited clone. But here what I'll do is only go through a few steps to kind of give you an idea of how we actually get the edited clone. And for sure you can, if you're still have more questions, you can definitely email me or look at the publication. But here I'll start with the second step where we essentially trap the cells from the heterogeneous population. We take that population, load into the syringe, and then flow it into in our device where it essentially traps the individual clones. We tried different flow rates, and as you can see sort of from our characterization is that, with 5 nanoliters per second as a flow rate, we can achieve the maximum efficiency of our device around 88%, which is great. Once you trap these cells, now you encapsulate with the droplet. The next thing you can do with our device is that you can have this on-demand functionality to release them anytime you want. And so here I show you a series of images from a movie that shows the actuation sequence to release the droplet on demand. So you can see the droplet is trapped. The 2 red dots represent the activated electrodes. Then you can essentially drag the droplet anywhere you want. And here is just kind of showing you what it essentially means. That here you can see the cell that's trapped in the region, and then you can release them using electrodes any direction you would like. By doing this, the key thing here is that you could release it through hydrodynamic focusing using a range of flow rates. You can see that it relies on the flow rates to have sort of this probability of likelihood of releasing. But having this sort of on demand, regardless of what flow rates you use, you can have this on demand release, which is essentially perfect, and it doesn't have to depend on sort of these flow rates, which is really nice. So essentially, if you want this clone, you can essentially release it anytime you want. Now, once you release the clone, the next thing what we want to do is actually recover it. So what we did was we followed this method from Johansson's group and published in SLAS Technology, where essentially you have this single cell droplet covered with oil. You put this on the PTFE device, mix it with media. The PTFE acts as a way to absorb the oil. And then you remove the oil from the droplet, and then you can take that droplet and place it into a well plate for further expansion, and then finally further verification. And so if we sort of summarize of what we've done, essentially what we can do is in terms of time is that we can actually knock out the specific genes. We can trap them, encapsulate, release, and then recover them, recover all these isoclonal cells in a matter of 45 minutes, as opposed to hours or months to actually perform this whole pipeline. And so if you want to do work on this full pipeline, you can use both of our devices, and it takes 1 week, approximately, I would say 1 week, to perform the actual transfection, selection, and sorting, isolating the single cell, and validating an expansion as well. So with that, I think I'll summarize my talk. I hope I've shown you that we're able to automate the gene editing pipeline using on-demand droplet-based microfluidics. We've expedited the workflow to less than 1 hour for trapping, sorting, and recovering cells, usually approximately a week for the pipeline instead of a month. And at the same time, you can download our hardware and software for your own implementation, and this is available at our Share Bitbucket website. And at the same time, I had one of my former students has started this company called Droplet, and if they're trying to automate gene editing using microfluidic devices, and if you're ever interested in learning more, I suggest you definitely contact Hugo Singha, who's actually the CEO of the company here. And with that, I'd definitely like to thank some people. Definitely my first student, Hugo, who's not here in the picture, but he's now the CEO of Droplet. Angela, who's also did a lot of the cell culturing, and also Kenza Fatime, who did a lot of the work for the single cell isolation. And of course, a lot of my funding sources here. And please, if you're more interested in learning what our lab does, definitely check us out on our social media feeds. And also, if you have any questions, feel free to email me. And I look forward to discussing some of our projects with you further. Thank you.